Hello class, welcome again to our molecular biology part 2. So this is just a continuations of our previous discussions, which is part 1, the isolations of your nucleic acid. For today's discussions, we were going to highlight about the purifications and the quantifications or the different methods of quantifying your nucleic acid. For the first quantifications of your nucleic acid, we will discuss about the density gradient centrifugations. So what is this? The concept of this density gradient centrifugation is just merely the separations of your DNA from your RNA and protein. It is different from the buoyant density. So when we say buoyant density class, this is actually the measure of the tendency of the substance to float in some other substances. No, so this is like example, this is the principles of your density gradient centrifugations, that your RNA has higher density than your DNA, and then your DNA is higher density than the protein. And to say, if there will be a tube here, the RNA is most likely at the bottom layer, and the middle layer is your DNA, and the top layer is your protein, because they have lower density. No, so the concentration gradient can be established using a high-speed centrifugation. Class, you have to take note that when you're going to perform a density gradient centrifugations, there will be settings, or you should set this high speed of centrifugations plus the additional of your CS chloride solutions, okay? CS chloride solutions and your high-speed centrifugations will yield to a very good quantifications of your density gradient centrifugations. No? So this is a typical pictures of you on how we're going to perform a density gradient centrifugation. So as I mentioned a while ago, that the increasing CS chloride concentrations from here to the downstream. So we have here the measurement of our CS density, by here. You can see here that from the top layer, we can isolate already a protein. This is after a high-speed centrifugations, okay? So we have here the top layer is the protein, your solutions, the middle part is your DNA. And then at the bottom layer is your RNA. Therefore, we can say that your RNA has higher density compared to your DNA. Your DNA has higher density than your protein. So the density gradient centrifugations is actually widely used to isolate your DNA, RNA, and protein. So these separations phage DNA from a coat proteins. No, this is actually also commonly known to separate the chromosomal DNA from the plasmid DNA if the CS chloride saturated with additional of your ATBR, or also called as your ethidium bromide. This ATBR will help to intercalates into the DNA, which is unwind as more dyes being dyed. No? So being bind. No? So as the DNA is unwinding and the dye is intercalating, the buoyant density is changing. So here is that the plasmid, you have also to take note that if you have the intact, like intact status, your plasmid cannot able to perform or cannot be able to unwind. No, your plasmid and your chromosomal DNA have different buoyant densities, or they have variety of buoyant densities. So all the forms of your plasmid DNA can be cytomusically separated and isolated using this density gradient centrifugation. So with your plasmid DNA is being isolated in this way, that is very pure. This is the removal already from your CS chloride and your ATBR. You can this can be prepared for a large quantities for your experimentations, fragments. So this is the basic concept of your density gradient centrifugations with the additional of your CS chloride and your ATBR. So here we have here already putting the layer of your DNA solutions on the top. And then we have the density gradients of your CS chloride and your concentrated ATBR. It will be centrifuged more likely to 18 hours. And then it's high speed for 50 to 60 RPM. And then you can visualize this one using a UV wand. And then you can see here that the upper band, it consists of your chromosomal and open plasmid DNA, or the lower band is the covalently closed plasmid DNA. So how you're going to remove this one is using a syringe of fresh tube. No, you can purify the CC plasmid DNA by using a syringe, no, and then transfer it to the fresh tube. So another one is we have here spectrophotometry. I know you're already familiar with the spectrophotometry because we already discussed this one, especially in our clinical chemistry now. It's already familiar to you. So with this spectrophotometry applications, so with your nucleic acid quantifications or isolations, there are different, how do you call this one, absorption spectrum. No? For the nucleic acid, the absorbance maximum is actually at the limit of 260 nm. No, the concentrations of these solutions of your DNA can be calculated at the average or the standard of your absorbance, which is 260. Plus, you have to take note that if there will be RNA in your sample, it will not be possible for you to operate or conduct or measure because your DNA concentration will be overestimated, no, of your RNA. So you have also to take note here that your DNA associated proteins are actually one of the component of your solutions, which is containing the DNA. So most proteins have the maximum absorbance of 200 nm. No? So the presence of this protein associated with the DNA will produce the solars, which is about 200 nm, which is I mentioned on the spectrographic curve. So here we have here the solutions or the ratios of your spectrophotometry based on the DNA and RNA, which is the ratio of the absorbance of 260 and 280, no, of 1.8 and 2.0 respectively. Yes, yeah, and always remember that if there will be a contamination proteins, your absorbance will increase into more likely more than 280. Therefore, it will decrease the value or the ratio of your spectrophotometry. For the conditions of your double-stranded DNA, the concentrations could be calculated using this formula. No? So we have here absorbance of your 260 of 1 is equal to 500 micrograms of your micrograms ml of your double-stranded DNA, whereas your single-stranded DNA, RNA, or some of your oligonucleotide, the absorbance of 260 is of 1 is equal to 300 micrograms per ml of your single-stranded RNA. Again, do not interchange these figures or formula, no, between the double-stranded DNA and your single-stranded DNA, RNA, or your oligonucleotide. For basing of your UV-based quantifications, now we have here quantifications of your double-stranded DNA, RNA, or your RNA peak and your protein peak. For the using your UV, no, for the quantifications, now at the peak of as I mentioned, that your double-stranded DNA peak or RNA peak as the maximum level of 260 nm, whereas your protein will peak at the curve of 280 nm. No, for the double-stranded DNA, we have here the optical density or the OD of this double-stranded DNA of 1 at the absorbance of 260 nm at the 50 UJ of your ml of your nucleic acid. No, so for the quantifications of your nucleic acid, so if you're going to determine the purity with respect to the protein that we had, so we can calculate this one with the absorbance of 260 over the absorbance of 280 ratio. No, if you have pure DNA, which is ratio lowered by the protein, you have pure DNA, which is a absorbance of 280 over absorbance of 280 is equal to 1.8. This is actually a ratio of your lowered protein. Huh? So for the part 3 of our discussions, we were also going to discuss about the recovery of your nucleic acid fragments from the main point of your agarose gel. So this is also important for you to at least you have the background of how you're going to recover a nucleic acid fragments from the gels. No, so first recovery is called electroillusions. No, so what is this electroillusions? This is mainly wherein your DNA is being electrophoresed, no, and the band containing the fragment of interest is cut out or is eradicated or yeah, cut out niya. So the gel slice is being placed in a dialysis bag. So no, we have a gel slice that being placed in the dialysis bag containing a small volume of your suitable buffer. So this dialysis bag that already have the gel slice, no, is being placed into the electrophoresis tank, no, and then you have to switch on the electric field, no, so there will be interactions between the positive and negative charges. So when it will already set up, the DNA will go into migrates out of the gel into the surrounding buffer, but it is unable to leave the dialysis sac. So that's why we could recover now our fragments. So the DNA is being then recovered from the buffer by the ethanol precipitation. Then, of course, class, we always remember that if you're going to recover a nucleic acid fragments, you should always add ethanol because it's going to precipitate at the level part which you can extract properly or conveniently. No? So we have here, alternatively, a throw is cut into the gel just ahead of the desired band. So there's also another step on how you're going to recover the, aside from adding the ethanol or ethanol precipitations, no, you could cut through the gel just ahead of the desired band. The through is being filled with, there's a lot of buffer and there's a voltage that was reapplied, no, and then the buffer solutions, which is contained the DNA fragment, is actually removed from the through with a pipette. So you could use a pipette from this one. And then, of course, after which you could also use ethanol precipitation for the recovery of your DNA. The simple as this is, no? So you have here example, this is the graph, no? You have DNA that is being electrophoresed already, then you have band cut, the gel is sliced, the gel slice is placed on the dialysis bag tube. Let's say this is the dialysis bag tube, and then the bag is placed into the electrophoresis. I don't know if this is the, should possibly, this is the electrophoresis of the tank, no? And then there will be some charges, the positive and negative charge. Then the positive charges will attract the DNA, no, or the anode, no. So the DNA here, example here, the DNA will migrate into the gel on the surrounding buffers, and then you can measure this one already. So that's how, same with gel electrophoresis, but the thing here is you have your DNA is already electrophoresed, no. So we have another methods which called the glass melt. So glass melt is just basically the suspension of a very tiny silica beads. No, these beads will help to bind will help to bind your DNA. No, so the DNA will go into bind to the beads or your tiny silica beads. No, so the DNA since bound to the beads can be precipitated by using a centrifugation at the maximum level. I mean the high speed centrifugation. So when the DNA can be eluted from the beads with an appropriate buffer or a water, you can also put elutions buffer. No, the agarose is being sliced containing the cell fragment is being dissolved again in a sodium iodide and a glass melt suspension added. No, that's why I call glass melt because at the end, instead of putting ethanol, they put glass melt suspension to make it precipitate. So another one is called LMP agarose, called also known as the low melting point agarose. So what does it mean? Because we going to use the 65 degrees Celsius and the remaining liquid, the agarose gel will going to melt at the 65 degrees Celsius and the remaining liquid at the 45 degrees Celsius. No? So the appropriate band is being exercised. Agarose liquefied by heating and the DNA being recovered directly by you adding a phenol extraction. So here we just used to add a phenol extraction. So especially this is very useful if there will be a successive restriction digest that being performed, no? So this is very common applications already in the laboratory. So if your DNA is being digested with the first array or frags separated on the LMP gel, then we have also here the individual bands are exercised and the agarose is being liquefied. No, the second array is being done in the agarose solution. So there will be some contaminants in the agarose that can be inhibit to their action. So the higher the enzyme concentrations is being needed for these reactions. No, when you're going to when the digestion is already complete, then the gel solutions is loaded to the second gel and the fragments again is being separated. Another here quantifications is of course a widely used commercial kits now, which is available already in the laboratory. So it's an easy task already because you just have to, we have here DNA precipitation methods and which is commercial kits already available to quantify or purify your DNA from the gel slices. So you have just to first run an agarose or PAG or PAG. You have to identify band containing desired DNA, and then you have to cut the band out of the gel, melt the gel using a chemical or heat, and then you have to spin the column to trap your the DNA. No, so this is actually just give you some insights on what will be the possible methods that you're going to perform if you're going to isolate or you're going to recover nucleic acid fragments. Now, I know you will also encounter some of this in the laboratory, but at least you have, because laboratory should also focus more on the procedures on how you're going to perform this method, experiment, no? But for this, we are also trying to give you some at least knowledge, no, basic understanding about the procedures of the nucleic acid techniques, no. So I'm going to share to you some of the video, which is from, this is a commercialized kit, no, from AppNova company. So this is called a gel purification kit from the AppNova company. So as I mentioned that if you're going to use a very commercialized kit, it is just easy, fast for a molecular biologist and more convenient and a faster results, no? Could yield faster results. But of course, there's a tendency that it might not that so accurate because some of the like sample we have to take note about the the kit that they're using. Maybe the buffer is not working already or already expired, no, or already contaminated. The reagent is already contaminated. So please, if you are a microbiologist that is rotating, that is using commercial kits, please make sure to double check the protocols or the standards of your um commercial kits that you're using for the experiment. Okay, so another here we have here in the most efficiency of your recovery from the agarose gel. So in general, regardless that of any methods that being used on the recovery of your DNA from the agarose gels, that is very less efficient for a larger fragments because of course you just already to recover those nucleic acids from the agarose gel. So this is very less efficient to recover a larger fragments, no, for a larger fragments, because larger fragments may be already shared, no, or scattered due to this simultaneously binding to multiple plasmid beads, which is actually we're using. Then we also, that's why we can if you still want to, if you still really want to isolate or like recover the larger fragments of your DNA or nucleic acid, then the most appropriate process of this one is to perform your electroelutions. No, for the small fragments as well, you cannot recover by using only ethyl acetate precipitation. No, then the best way to recover this one is through a glass milk procedures. Again, if you want to recover a large fragments of your nucleic acid from the agarose gel, the best solution is using the electroelution. However, for the small fragments, since you cannot merely recover this one to the precipitations of your ethyl acetate, then the best option is using your glass milk procedure or glass milk beads procedure. Okay. So for the part 4 of our discussion, we're going to discuss about the analysis of your gel electrophoresis. So as you can see, this is always like widely used in our laboratory. And you can see here, if you're working in a laboratory, you could always check this on how to analyze your gel electrophoresis using this graph or using this kind of results. No? So what is gel electrophoresis? What is why we need to use gel electrophoresis? So gel electrophoresis is the purpose of this one is just to purify the nucleic acid as well as the proteins. And then one of this one also is used because we need to identify which specific DNA molecules that have been isolated. No? Sa itong mga DNA molecules that we already isolated and already cut by some of the restriction enzymes. Because of course, we're going to perform another methods. We also perform restriction enzymes, and then we need to isolate unsa man tong DNA molecules, no, ang na-isolate o na-cut by the restriction enzymes. And also another purpose of your the electrophoresis is determinations. Now, if we're going to determine the different of your genomic of your different plant and animal species, this is very very important. No, gel electrophoresis is always and widely used in the laboratory, molecular biology laboratory, because if you want to identify the genomes of structures of the different plants and animals, then first step of we can perform is gel electrophoresis. No, and another purpose of your gel electrophoresis is, of course, if you want to compare a DNA fingerprints, no, which is example found in the forensic crime, no, and then that is subject for is forensic science. So your DNA sample could be collected from blood or semen are being separated using your gel electrophoresis. Then the number and the positions of the bands being formed on the each of your lane of your gel is the actual DNA fingerprint and is used for that is very unique for each person. So see how very important for us to at least determine what to know at least the basic understanding and the purpose of your gel electrophoresis. So gel electrophoresis is the gel matrix that is called jelly-like porous material that supports and allows a macromolecules to move in. That's why called for electrophoresis, perhaps using some of the charges. But here we have your gel electrophoresis. This is a gel-like porous materials. No, this will help to support or move the macromolecules, no, all throughout from lane to lane, no. So but there are two different gels matrices that we are using in the laboratory. One of the most common like called gel matrices that in the laboratory that's used is the agarose gel. However, here some of the others laboratory also using polyacrylamide na, gel matrices. But for us, we use 0.25 or 25% of your agarose gel added with other solutions. So the DNA is we all know that the DNA is a negative charge because of we have the phosphate background, no? The DNA will be attracted to our positively charged poles and then being repelled from the negatively charged ones, no? So most commonly, gel electrophoresis matrices that we use, and as I mentioned, is for the DNA. If you're going to use DNA agarose, I mean, if you are going to use matrices from agarose, then your purpose are identifying the DNA and the large RNA molecules. But if you are going to use poly, then you could use with or without SDS for proteins, no? Or with or without urea for RNA and small DNA molecules. So this is a typical picture for you for a gel electrophoresis. Now we have here the how they call this machine, the charges, and then we have here just this is the turn on the switch on, switch off machine. This typical one. So we have the electrode and electrode, positive and negative electrode, and then we have here the DNA fragments and the agarose gel, which is put here. And then we have here the buffer solutions. This is the, we can put a lot of buffer solutions here. It should not at least at the label na lang siya sa standard, no, sa maximum siya, di dapat siya mag-overflow. And then we have here some of the standard fragments move. So once it will return on, so the small DNA fragments is being moved farther to the gel than the large large fragments. So for the agarose gel, that we were going, this agarose gel is, as I mentioned, is widely used in the laboratory. So the migrations rate of the DNA in the agarose gel depends on the different factors, of course, no, because the purpose of this one is to migrate the micromolecules, no, to move through to the specific lane. So for the one factors of these migrations of your agarose gel of your DNA is first is molecular size. No, the rate of the travel is inversely proportional to the molecular size. Another here is the DNA conformation. So we have here DNA conformations, the shape of your the DNA. No, so we have your supercoiled, relaxed, cellular, and linear DNA of the same mass will migrate at the different rates. Now you can identify here if they go, or this is super cold, this is relaxed cellular, or this is linear DNA, because they will going to move through the lane at the different rates. Another one is the concentrations of the gel. So as I mentioned that somehow we use also 0.8% of gel, of agarose gel. Other will use 0.25% of the agarose gel. So it depends, no? So the concentrations of the gel will also affect to the fact of the migrations of your DNA, no? So it provides separations of 0.8 to 10 kb range, no? Another one is the electric field strength. So it's already obviously that if you have a low voltage of the rate of migration is very proportional to the voltage. So if you just use very low voltage, then it will also travel very low or very slow, no? So this is how you're going to prepare for the agarose gel. So this is the agarose gel, which is commercialized which already from the company. It depends now. You have to read the protocols, which is the insert of this agarose gel that you bought from the company now. So we have here the agarose gels that have fairly large pore size, so are used to separate the larger DNA or the RNA fragments. So basically, literally, no, it is running a gel because of course you need to run for a gel. So it's used that after your electrophoresis, you said people said, oh, I have to run for a gel, meaning to say you have to run your sample in gel electrophoresis. It's a term in molecular biology or even the laboratory. So called molten agarose is being poured into the casting tray and the comb is being placed at one end. So it's this is actually just insights for you because I believe in your laboratory, your professor will going to help you to demonstrate no of how to perform at least no sa laboratory no they were trying to give you some insights what will be the basic steps. So this is just overview for you. So we have here molten agarose na inibutang sa casting tray, and then there will be some comb, how to call this one, kind of layer by layer na mura siya kining comb gyud, no, that will separate na specific size from that. And then after the agarose gel, so you have to wait around 30 minutes, most likely 20 to 30 minutes to make your agarose gel solidifies, no. And then the comb is you gently remove it, no, dili dapat ninyo na magpaspason pag-remove because it will damage the gel, no? So just be gently and carefully remove it. And then after which you can mix the DNA samples if you already prepared your DNA samples, which mix it with your tracking dye. So we have first if you're going to run a gel, of course you have to take note that first lane you have to put a tracking dye, no? Which is like how do you call this one in we call it also like control, mura control nimo siya. So usually it colors purple, no, purple color, no. First lane gina siya just to identify, no, that you have a good samples, no, na magputang control na. So usually if your DNA samples with mixed with the tracking dye, which is contains with your sucrose, and then the dye so that you can also visualize the migration because after this one you have to scan this in the UV light, no, and then you can take a picture with that. So this dye will help you to visualize if there will be a migration. So this sucrose, the purpose of this sucrose is to weigh down of your DNA. And then after this one, a buffer containing ions to conduct electric current is placed in the chamber around the gel. So hopefully, no, your laboratory professors were able to give you the insights of how you're going to run a gel or some video that could help you to run a gel. It's just an easy test, but you have to be careful because you're dealing with electricity or electrodes of this one. And also, plus, I have to mention here that some of the dyes, especially the tracking dye, is cancerous. So you have to be careful. No, we have to be careful, especially the ATBR also that we'll be using in the gel electrophoresis is also cancerous. So you have to be careful not to avoid contaminating now, or I mean have this the contact directly to the reagent that you're going to use in the electrophoresis. So in our laboratory, as I rotating, I mean as I'm doing the gel electrophoresis. Oh, sorry, this is the comb that I mentioned previously, no, Marisha Gani. And then you put your gel here. Then you have to, yeah, after you put your molten agarose here, then you put the comb. This is kining yung mga gap niya, which is actually this is the measurement na kung pila ka measurement sa imuhang sample. So if you want large fragment, you could use this one. With small fragment, you could use this one. No, it depends on your preferences. No, so yeah, after this one, it will solidify that the result will be this one. So gently remove it. No, after you have to put this one in the buffer, but in the gel electrophoresis box. Now carefully, then you could start adding first phase of your tracking dye. And then, yeah, as I mentioned, you have to be careful with this. So when performing a gel electrophoresis, you have to take note that you have to avoid contact directly to the reagent, especially because these are some of the reagents are using the gel electrophoresis are very cancerous. So if you are like you have gloves, ang gloves dapat gamiton ani is intended lang yun, or ang pipette nga gamiton is intended lang yun sa gel electrophoresis, no? So if after nimo perform gel electrophoresis, then you have to trash again your gloves, no? The pipette that you're using is intended lang yun, di dapat siya gamitan for general procedure. Kung nada may pipette nga intended or tips nga intended lang sa gel electrophoresis, no? So this is how we're going to, this is the concept of your gel electrophoresis. So we have here positive and negative, positive and negative electrode, no? So this is your agarose gel. This is the DNA fragments that you use, and then we have your buffer solution. So once it was turned on, so there will be migrations of your DNA, no? Using the dye, no? So as you can see here, the small DNA fragments move farther because they are small. They can move farther to the lane. However, the small fragments are like sample, as I mentioned, there are different characteristics or factors about the migrations of your DNA or your samples, no? It depends, no? So we have here the larger one will migrate only like slow, or even like dili ana siya kataas yung pag-migrate. So we have also a well-known page, no? This is page, I'm using page since like I was just introduced page or gel electrophoresis last year, when we're going to isolate the bacterial strains for the, we're going to isolate for the E. coli. So always may nagagamit ani gel electrophoresis page, no? For for your page gels, no, it can be manufactured with a very small pore size. Gamay lang kayo siya. So the length differences of one base pair can be visualized by cold page, no. So these sequences is carried out by at least 6%. Usually, yeah, 6 to 8%, let's say. But in the references we have, it's 6% page. So the urea is being included just to help to eliminate the conformational effects, no? And then 2 to 10% of the page can be, plus you have to remember that if you have 2 to 10% of the page, it could help you to separate already your RNA. Your SDS page is widely used for the separations of your protein. Again, your SDS page is widely used to separate your protein. So this gels can be stained with a composite brilliant blue or silver, no, stain to visualize the products. So in our laboratory, we use silver stain, no, but some of the laboratory they use brilliant blue. So it depends on the preferences that they have or the product that they have. So this is to help this is to help to visualize the migrations of your products, no. This DNA is often directly labeled for a visualization. So this is what is the settings of your page, no? Like ito siya sa electrophoresis. Same lang sa settings, pero ang dili nakadapa siya, ang dili sa pikas nakatindog siya nga na siya layer. So we have here electrode. We have here positive and negative electrode. We have positive electrode. So you could put here same agap sila kung na siya process, no? So you have to put here your gel. This is your gel, the blue one. So we have here very small fragments, and then the upper part is the larger fragments, and then this is called the well. You can put your samples here, and then yeah, so this is the results now. So at the upper part, you could see that this is the high molecular weight protein, and then this is the protein band. You can see very continuous now, high molecular band, protein band. And then this is the smaller fragments at the lower part. So the directions of the protein migrations is from the top to the bottom here. Now you can see here. So if nasa sa taas ang imong makakita kang sa imong result is napa sa taas ang band sa taas, mean to say, oh, these are the larger fragments, no? And then if it will break more farther, then it could be a smaller fragments. It's just easy, same methods of the gel electrophoresis, but they're using different solutions here. So how are you going to visualize? Because we're always talking about visualizations of your DNA. How after running this job, of course, you need to visualize your DNA or nucleic acid. So of course, using your ATBR, no, or ethidium bromide. So again, this is very, very cancerous as well. So you have to be careful of, soaking or getting subsample this one to your gel electrophoresis. So we have here, this is a fluorescent dye visualized. So you under the UV light, it will appear like blue or sometimes red, sorry, blue or somewhere purple, no? When excited by, so it will intercalate into DNA molecules thereby staining it. So since it will intercalate in the DNA molecules, mag-reflect siya sa UV light. So you can see this one. So the gel is being soaked into the solutions of your ATBR, and the DNA bands take up the dye, no? And then the gel is being placed under the UV light, and then you can photograph this one. So now may machine ana, you can just insert your gel into that, then you have to scan under the UV light, then you can adjust the saturation, the brightness of that, and then you can take a picture, photograph, print screen lang. Automatically na if you have already machine. So mo siya ang picture, sorry, or you have also here picture, photo prep. So you can put here, see, you can, because of the ATBR, it is like reflecting, no? Nag-stain siya with the help of the ATBR. So that's why this is how it looks like, no? So this is the machine, but I'm using not this machine, I'm using the big machine. This is just old machine, no? Pero dali lang siya nga machine to use. So, but yeah, this is the photo prep for the visualizations of your gel electrophoresis. So now we have here the sample from the scanned or photograph under the UV light, no, using a ethidium stain gel. So you can see here that, oh, okay, because this one's supposed to be the tracking part, no, but yeah. So here is we have here at the, okay, you can stay here that this part are the larger like molecules, or like this part is the, let's go here, example. Okay, we have here another, this part is the larger fragments. So this part is the lower fragments. But how can you identify good, no? Just to see this one, oh, this one is the larger fragments, but we don't know how much. So that's why there will be a measurement of that one, how much QB ang naka-assign dira. But if you want to confirm it really, you could use also a nanograph. Nanograph is a measurement of your DNA fragments, no? So you could also use to confirm, but for me, I always confirm this one. After gel electrophoresis, I measure the quantity using a nanograph. No, it's really more accurate for me. But of course, you cannot do it without also performing the gel electrophoresis. So the movement of your DNA fragments in agarose gel, see here. So here is your larger fragment, and then here is the lower fragment, no? But we need to have our references beside to compare, no? So we have here the movement of your DNA fragments in the agarose gel. So there's a linear relationship between the migration rate of the given DNA fragments and the logarithm of the size, no? It depends, no? If you have bigger size, then it will move slowly. If you have smaller size, then it will move farther from the main point. Then we have here the relationship is used to determine the size of the fragment from the gel photograph. No, so of course, this is actually references for us to determine that, oh, this product or this DNA product in sample number one, sample this one, have like usually they have like larger product, no. So you could compare this one. This is really perfect. This is also perfect one, that it has at least, no, you can visualize this one properly. But of course, as I mentioned, we have the standard measurement from this, which is I can show you, no? So later, so we have here the size estimation of the DNA fragments in the agar cell. So you can measure the distance migrated from the original point, from the well, the DNA or RNA or protein bands of the marker. So we have a marker, So the accuracy will depend, of course, it will depend on the accuracy and the consistency of the measurement. So this is how you're going to compare the band. So sir, saan man naman na pag-measure, sir, if you have the agarose gel, then you can compare using this band, no? This is Patrick na again. But in the laboratory, I believe that I'm hopeful that your professor will help you to interpret properly or maghatid sa example how to interpret properly your agarose gel, no, results. But this is just the concept for you, know. So you have to compare the band with the marker used and you'll be able to figure out the length of the fragment. So example, this is the band you run. This is the standard DNA length, no? So we have here positive sample A, positive sample B, no? Let's say that one. So this is as I mentioned, this is the tracking point. No, the first should be at least the tracking point. So this is 10,000 base pair, 8,000 base pair. So you can see here, no? You can see the measurement. The lower part, of course, there are bigger size, bigger density, bigger maybe the shape one, because they migrate upon like after the process of electrophoresis. They still in the upper part, no? You can see here the base pair, but this is the standard as I mentioned. Yeah. Okay, and the lower part is a little blurry because they are very smaller, no? Then we can have here is positive control sample A. Example, this is our positive control. So the positive control of your DNA has a base pair of 750, no? Base, you can always compare the size. There are some instances that when you're going to run your gel electrophoresis and then you have the standard length, but your positive control is half lang siya or your sample is blurred siya or gamay lang siya nga fragments. Do you mean correct ba siya or dili? So there are some instances, there are factors that happen that, so mean to say you didn't properly put the sample or there will be a problem or there will be contaminations of your sample. So in our cases, we need to repeat it or we need to confirm it, or because our professor our when you're going to present this one in the public, people say, oh, you run the gel improperly, no? Dili mao imong pag-run sa gel. So it's invalidate the results as well. So we need to rerun it or we need to measure it using a nanogram or nanodrop. Sorry, not we use it nanodrop. So, but in these cases, it's really nice because they have the same shape, almost the same size, no? And the deflection is so nice. So mean to say the positive control of your sample A is in the 75 base pair. However, here, we have here the specific test for your sample A. You can see here below down, which is you can still see at this point. So I don't know, but at the other references, they still have also below 250 base pair. So you can measure that as nag-depend sa measurement na nila, no? So of course, with your DNA, or I mean your negative control sample A, you also have to put negative control. So negative control, of course, wala dapat kayo ma-isolate rin ng DNA because it's negative, of course. So this is species-specific test sample A. So they have below base pair of 250. So these are the additional learnings or resources that if you ever still misunderstand, I mean, if you have confusions of understanding or analysis of the gel electrophoresis, then I advise you to read some of the resources, or I also need to upload this one in the Canvas. So you could also read this one for your references because of course, we don't have much time to discuss everything about gel electrophoresis and then basic nucleic acid, because I believe this is just a supplementary for you, a theory for you to back up your skills in performing the laboratory in the molecular biology. And I believe that it will be discussed properly or more like deeply the detailed procedures on how to perform a gel electrophoresis or basic techniques in the molecular biology lab. But however, I just want also you to not to rely everything to the discussions. That's why I'm giving you some online resources here and here. And if you have time, you can read it properly. And yeah, and if you have concern, you can email me about this one. So for the textbook that I'm using the isolation DNA and RNA. And then also I'm using the techniques for molecular biology by Trop BA. So again, thank you so much, guys. If you have clarifications or like concerns, please don't hesitate to email me. And God bless and happy Montan.